Welcome to our presentation on machine learning-based quantification of ovarian follicle stages and heterocephalus Glaber. I'm Pamela Styborski and my teammates are Julian, Martin, and Silvia with Arizona State University.
Hi, I'm Martin Orkuma. So to get started, we have to address the bottleneck in reproductive histology, which is manual follicle identification. Right now, counting and classifying follicles is incredibly labor intensive, and it relies on the individual observer. And frankly, it's difficult to scale for large scale research. So what are we doing differently and why now? So we've reached the perfect storm of factors. For example, we know that histology is now done digitally, and CNNs have become very sophisticated, very capable of analyzing large scale data. Plus, we have repositories such as the Mother database that provides ready to use high quality data to the public. And then we have the naked mole rat, which is an ideal biological species that no one has built an automated follicle identification system for. So our goal here is to combine these factors with ready-to-use slide repositories, to combine that with modern machine learning to fully identify and classify ovarian follicles. Our primary objective is to determine if a machine learning pipeline can effectively replace manual counting in Glaber ovarian histology. It's about moving from a human-dependent estimation to a machine-led precision. So we've identified three gaps so far. First is the scalability issue I mentioned earlier. Secondly, while the naked mole rats are a premier model for studying reproductive longevity, we simply lack the automated tools to study its ovaries at scale. And then lastly, while the Mother database has done a fantastic job in providing images and data, it doesn't provide a reproducible workflow to process that data or those images. So, our specific research question is, can a ResNet 34 model trained on tiles we've annotated in QPath accurately and effectively classify follicle stages and then aggregate those local predictions into reliable slide level counts? Our hypothesis is straightforward. Machine learning can automate the identification, classification, and quantification of ovarian follicles in the naked mole rat, essentially replacing manual effort with a scalable, reproducible pipeline. To test this, we've broken automation down into four distinct functional steps. You can see the steps in our workflow below. The first step is identifying. This is the detection phase. We're isolating the actual tissue regions within the massive whole slide image. Next, we go to classifying, where we assign every individual tile to one of 8 specific follicle classes. Then we move on to quantifying. This is an aggregate step where we turn those thousands of tile level labels back into a meaningful count for the entire slide. Lastly, we go to automating. This is the final step. The goal here is an end-to-end process where after the initial training, the pipeline runs without any manual intervention.
The image shown is our workflow pipeline graphic. We begin with our data acquisition pipeline, starting with tissue collection and periodic acid shift staining on the slides. Slides are digitized and included in the multi-species ovarian tissue histology electronic repository. We break down the dataset composition and examination of the XML file characteristics. The pipeline then walks through data processing, focusing on normalization, tissue detection and enumeration, and data augmentation. Moving to model training, we focus on slide annotation and the central neural network. The pipeline concludes with model inference and the evaluation of the model, including the confusion matrix and the ROC curve. The image shown is an export from Python after cleaning and breaking down the dataset. Digital histology slides and structured metadata were sourced from the multi-species ovarian tissue histology electronic repository known as Mother, with funding from the National Science Foundation. The dataset comprises 9 individual naked mole rat donors representing diverse life stages. 6 neonatal juveniles aged 1, 5, 8, 15, 28, and 90 days, 1 prepubertal juvenile at 180 days, and 2 adults at 3 years old. All tissue samples were derived from the left ovary, sectioned at a thickness of 6 micrometers, and stained via periodic acid shift.
Hi, my name is Silvia, and I will be covering some of the exploratory data analysis. To get familiar with the tissue in our data set, we used some tissue detection and cell detection within QPath to run some EDA tests. We were looking at the eccentricity and the circularity of the cells. We were looking at how nuclear size varied across our sample. To do this, we used a method called the kernel density estimation, which creates smooth curves to represent some nuclear areas, how the nuclear areas are distributed. We also normalized the data so that we're focusing on shapes of the distribution rather than the number of nuclei in each sample. What we see is that most of the curves overlap quite closely, which suggests that the nuclear size patterns are fairly consistent across the slides. There's some skewness present, meaning a slight imbalance is in how the values are distributed, but overall the trends are very similar. We also confirm these findings using a box plot, which showed comparable patterns across all samples. Here I'll walk you through how we generated the data for our analysis. We analyzed whole slide images using QPath, which is designed specifically for histological image analysis. From the 9 slides available from on the naked mole rats, we selected 3 for detailed manual annotation to create a reliable reference dataset. We started with automated tissue detection to identify the relevant regions, and then trained a classifier to segment different tissue types, as seen here in the visual with the pie chart representing some of the follicles as being detected. For consistency, we followed a standardized labeling approach for annotating these structures. Once the images were annotated, we generated smaller image tiles from those regions, as you can see in the visual with the tiles. This is an actual output from QPath for our dataset. These tiles maintain high detail in structure, which is a QPath quality, making them ideal for use in downstream machine learning models such as ours.
To design our machine learning pipeline, we implemented a ResNet34 convolutional neural network to analyze our naked mole rat ovarian histology slides. First, we used the whole slide images annotated in QPath to label follicle classes such as primordial, transitional, primordial, primary, multilayer, antral, and atreidate antral, along with stroma and germ cells. From these annotations, we extracted labeled image tiles. These tiles are split into training, validation, and test sets recognized by the ResNet34 model, and the model is then trained using transfer learning that allows the ResNet34 to learn distinguishing histological features efficiently while monitoring performance and saving the best model using callback features. For evaluation, we applied the trained model to whole slide images by dividing the images into appropriately sized tiles. Each tile was classified by the CNN, and predictions were grouped across the slides. This produces class-specific counts and overall follicle distribution, enabling automated scalable analysis of the ovarian tissue and supporting quantitative and some age-related insights into follicle development and tissue composition. Some of the results from the training and validation of our CNN were checkpoint outputs that relayed the validation loss and error as well as the test error. The best validation performance was obtained during the frozen phase of training with a validation loss of 0.783. This cross-entropy loss value would indicate moderate performance, where the model is making reasonably confident predictions, but there is still noticeable error and some uncertainty. Our model achieved a validation error of 26.2% and a test error of 32.1%, which again aligns with this moderate performance and highlights some important limitations. The increase from validation to test error indicates that the model may not generalize perfectly to new unseen data. This could likely be due to variability in staining, tissue structure, the limited tile volume within the trained CNN folders, or class imbalance. Looking at the confusion matrix here on the screen, we see that certain follicle classes, particularly those with similar morphology, are more frequently misclassified. This tells us that the model is learning meaningful features, but still struggles with subtle developmental differences. These results seem to be quite typical for histology-related tasks, where visual distinctions can be nuanced even for experts.
A receiver operating characteristic curve was generated to evaluate discriminative performance across all 8 classes. The graph shows the true positivity rate of the model compared to the false positive rate. All classes achieved area under the curve values substantially above the random chance baseline of 0.5, with a mean area under the curve of 0.93 across all classes. Per class area under the curve scores were as follows: transitional primordial with 1.0, primary with 0.99, germ 0.98, marking the 3 best classes. Antral area under the curve was 0.95, primordial was 0.93, and stroma was 0.91. Our lowest classes were multilayer with 0.86 and atriac antral with 0.84.
Once trained, validated, and tested, we used all 9 naked mole rat slides for mother to evaluate the application of the CNN to whole image ovarian histology slides. To be evaluated by the CNN, each slide was tiled into non-overlapping 224 by 224 pixel patches, and a mean intensity threshold of 220 pixels was applied to exclude background tiles. Here we have 2 different visualizations of the distribution of predicted follicle counts across all 9 slides. The stacked bar graph shows the percent composition for the different classes identified on each slide, while the heat map enumerates the tile count per class across the slides. As you can see, the adult naked mole rat slides, ascension numbers ending in 37 and 38, have a higher number of atriatic antral and antral tiles when compared to neonatal and prepubertal slides. This is likely due to the more active and rapid folliculogenesis observed in adult mammals, as these 2 follicle classes are considered later stages of development. The neonatal and the prepubertal slides have more diversity in their breakdown composition, which could be related to the varying ages that range from 1 day old to 180 days old, while both adult donors were recorded at 3 years old.
Hello, here I will cover the discussion part of our study. When it came to model performance, the ResNet 34 achieved moderate classification accuracy overall, with its best performance during the feature extraction phase, where the validation error reached 26%. This suggests that the pre-trained features were already well enough suited for capturing the key morphological characteristics present in our slides. When we moved into fine-tuning, performance declined rather than improved, which was interesting. The model became too overfit, which indicates that additional training may have disrupted useful feature presentation instead of refining them. In this case, the pre-trained network appeared to be best for our goals given the dataset. This was also supported by the drop in performance on the independent test set, where the error increased to 32%. This gap between validation and test performance highlights limited generalization. Perhaps this was due to the small dataset size, the variability between samples, and the class imbalance. Looking more closely at class level performance, the model performed best on well-defined and visually distinct categories, such as the primordial follicles and the germ cells, which were found in our young specimen. In comparison, the performance was lower for more complex classes, including stroma, multilayer, and atretic antral follicles, which are found in our adult specimen. Most misclassification occurred between the biologically similar and developmentally adjacent stages, suggesting that while the model captures general morphology, it struggles with finer distinctions. For the relevance of our study, biologically, the model output remains meaningful despite the limitations. We observed clear age-related patterns across the whole slide predictions. Adult slides showed a higher prevalence of antral and atretic follicles, while younger samples were dominated by early-stage follicles. These trends are consistent with non-ovarian development, which supports the idea that the model is capturing real biological signals rather than random patterns. For reference, the visual highlights the follicle stages as follicles move from one stage to the next. The follicles with the most complex structures were harder to identify with this study, but were accurately found within the correct slides. As for limitations, several limitations are considered. The dataset is small and heterogeneous, which restricts the diversity of features the model can learn. Class imbalance and annotation constraints also likely contributed to instability in certain categories. Additionally, the tile-based approach, while it is efficient, does limit spatial precision, and may reduce accuracy in regions with dense or overlapping structures, such as those in the adult specimen. This points to an important trade-off in computational histology, comparing deep learning versus machine learning approaches, in which the amount of data that is used are quite a big gap. The tile-based methods are scalable and they are practical, especially for smaller data sets, but they do sacrifice the anatomical detail compared to segmentation-based approaches, which provide more precise localization at the cost of increased complexity and annotation efforts. Overall, our study demonstrated that automated histology analysis of the naked mole rat within mother dataset is feasible and can produce biologically relevant insights. However, improving the robustness and generalizability will require larger and more diverse datasets, more detailed annotation, and potentially a hybrid approach that combines segmentation, which would identify the exact boundaries of each follicle. And this combined with classification is a promising approach.
Thank you for your time, and here are our references.